All right, what's up? What's going on? Yep, it's me again with another video that is gonna detail my travails into the search for eternal life. If that was too many words for you, then I'm sorry. You might notice that I'm in a room, right, that has a lot of echo in it. Yeah, I'm in the bathroom. Okay, cool. That's where we're gonna do these lessons and where I'm gonna do my research from now on, okay? So let's get started. Last time I talked to you about microtubules in the cytoskeleton, all the cytoskeletal structures, okay? And remember, there's tons of cytoskeletal structures. There's three primary categories that everybody is familiar with in high school biology, which is pretty much and this is just a recap, so we're talking about the cytoskeleton, right? And underneath the cytoskeleton, you have the microtubules, and you have, actually, you know what I'm going to do? I like MS Paint Online. This is an awesome program, but I'm going to actually start up Tor. Here, I think that's a better idea, right? Tor browser. There we go. Let's get that Tor. Oh man, I gotta restart Tor to update it. Okay, who cares? Tor circuit established, blah, 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 blah. Okay. And I have a reason for starting up Tor, okay? Because Tor is freaking awesome, right? Okay. Here we go. Okay. Now Tor's got the window. Let's remember to do this next time, okay? Paint online. This should get me to that JSpaint app. I like JSpaint. Where is that? JSpaint online. There we go. JSpaint is awesome. Hopefully I can actually use it. Oh yeah, I can use it on Tor. Nope, I don't want it to access my data. Why would I need JSpaint to access my data? Okay, let's go back to what we were talking about. Yeah, we were talking about the microtubules. Microtubules. I'm sure I spelled that wrong. And if I go, there we go. If I go to Wikipedia, you get all that stuff that you need there about microtubules, okay? But we typically don't go to Wiki for classes because, yeah, why would we do that? You know, you could go to Wiki, but some people, you know, you don't get as much from just reading a Wikipedia article, do you, as an actual lecture. So that's what I'm going to try to do for you here. So remember, microtubules. What are microtubules? Microtubules extend mostly from the nucleus, right? The nucleus is here. It's the center of the cell that contains all the DNA. The nucleus, and this is going to be relevant later, is composed of a double phospholipid bilayer. Okay, so it's a bilayer, like bye-bye birdie, like bye-bye, like Trump would say, right? And anyway, back to microtubules before we go back into the nucleus, because the nucleus is going to be important for the thing I'm going to talk about. But remember, microtubules are the cytoskeletal structures which are responsible for transport of various things throughout the cell. Okay, those things could include proteins, entire virus particles, you name it. And I'm going to find out later a little more about the details of what exactly goes through microtubules. But for now, we just want to know that microtubules are pretty big, right? They're the biggest cytoskeletal structure despite being called micro, okay? There's not, in the relative scale of things, there's nothing micro about them, okay? And microtubules are about 25 nanometers in diameter. I guess that's something like that. We can double check. Microtubule average diameter, okay. Let's see, 23 and 27 nanometers. Well, the inner diameter is between 11 and 15 nanometers. Okay, so there's a difference. The inner diameter, 11 to 15, right? That's this. Oh my goodness. Is there a better tool? Okay, I just want the little tiny thing. Oh gosh. I should have just expected this from the online paint program. So inner diameter this is not the scale, is 11 to 15 or 13 nanometers, the outer diameter. And oh gosh, all right, we're gonna get this right? This is how we're gonna do this. We're gonna do this from now on. We're gonna label what we're gonna talk about. Okay, microtubule. Okay, microtubule is I'm getting my tube-like thing ready here. All right, and that looks like this. And we have our inner diameter here, we're saying. Again, not the scale. Outer diameter. Okay, outer diameter here is about 23 to 27 nanometers. And other cytoskeletal structures, we have our microfilaments, which are composed of actin, right? And microfilaments usually are pictured outside around the cell boundary, right? They are like right around here. And I might, like when I think about microfilaments, I have to do more study on them, but they provide a support structural role to the lipid bilayer of the cell and other membranes of the cell. They provide structure to the cell, right? And they resist compressive forces. So it prevents a cell from collapsing within itself, okay? Which is very important because remember, you have water outside, you have water in the cytosol, which is the fluid in the cell. And you have a pressure gradient from outside the cell to inside the cell. So you need different resistive structures within the cell to prevent the pressure from destroying the cell, which is one of the things the microfilaments do. Microfilaments are composed of actin, right? And actin, I don't know too much about the actin protein, but actin and myosin often go together, and we were talking about the other day motor proteins, right? Myosin's a motor protein, actin is the structural protein, right, that comprises the microfilaments. And then we have after actin and in microfilaments and myosin, right, we have what's the largest thing. The second largest thing is called the intermediate filament, and in this case it's aptly named intermediate filament, and therefore being aptly named, it should have a larger thing with a different color. I'm going to make them blue. Intermediate filaments give tensile strength to a cell, and it resists tensile forces. So basically, when cells are stretched, the microfilaments prevent them from being torn apart, which is something that happens quite often, right? I mean, under different conditions, like for example, when you stretch your skin cells, right? Your skin cells have to resist a lot of tension, right? Because often you're stretching your skin, right? So microfilaments in the form of keratin they're composed of keratin. Not microfilaments, intermediate filaments are composed of keratin, and they prevent your cells from tearing apart. Okay, so remember compressive forces for microfilaments, intermediate filament, tensile resistance to tensile forces for intermediate filaments. Okay, and where all of these, remember, are composed of different things. Microtubules are composed of tubulin, right? Which you have an alpha subunit and a beta subunit. And intermediate filaments are composed of keratin. All of these structures also I'm not 100% sure of yet, but I will eventually. And actin composes the microfilaments, okay? Actin monomer, all of these are monomeric, or units that are combined together into polymers, and these are like long polymers. In fact, in a certain way, they can be, I guess, considered as macromolecules, but they're not considered as macromolecules because macromolecules are the ones that are used pretty much universally throughout your system for different functions, like carbohydrates, lipids, amino acids. Those are the universal macromolecules of your system, or those are universal Proteins also, not amino acids. Amino acids comprise proteins and DNA. Remember, there's 4 macromolecules in your system. Okay, so the next thing is, let's get to what I wanted to talk about today mostly. Okay, again, this is haphazard, so if you're watching this, you might be like laughing. You might be like, wow, this is all way out of order. Yeah, well, you know what? I'm on the search. I started my search for eternal life just from the very beginning. Okay. And I will include a few minutes here about like more than just a biology lecture or, you know, high school / AP Chem or AP biology lecture. I will go into what's the current state of eternal life, okay, including things that David Sinclair talked about, Elizabeth Blackburn, telomeres, all that stuff. I'm going to be doing my research there, so maybe we'll research together or something like that, okay. Either way, so this is the nucleus of a cell, right? The nucleus, right, has, and we'll label this nucleus of the cell. The nucleus is composed of a bilayer, okay, double bilayer. So not just a single phospholipid bilayer like your cell membrane is composed of, but a double bilayer, okay, the inner nuclear membrane and outer nuclear membrane, okay? This is inner nuclear membrane, outer nuclear membrane, okay? The difference between the inner nuclear membrane and outer nuclear membrane, I mean, there's probably a ton, but I'm not going to talk about them all right now. And then within the nuclear membrane, there's channel and transport mechanisms which utilize these nuclear pores here. So why is there transport here? Because obviously mRNA has to make itself outside of the nuclear membrane, and the only way that mRNA is going to do that, because remember, what do you remember about the phospholipid bilayer? The phospholipid bilayer consists of, these are phospholipids, right? Remember, these are diglyceride. Phospholipids are diglycerides. At least the nuclear phospholipids are diglycerides, right? And you have your fatty acid tails, right? One of them has a kink in them, so they kind of fit into each other, loosely speaking. And your polar heads, polar hydrophilic heads, okay? And this is the micelle structure, right? You know what the micelle is. Oh, the micelle is basically what would happen if you threw a bunch of phospholipids into water. They form micelles because of electromagnetic polarity, the hydrophobic ends kind of clump together. The and they form these chains. These fatty acid chains are hydrophobic. These phosphate heads, right, are hydrophilic, and therefore they face the water side because they like interacting with the water. The hydrophobic ends don't. The reason I'm bringing that up is because mRNA is essentially, what is mRNA? It's a bunch of amino acids stuck together, and so they can't just exit through this hydrophilic part here because they won't just give access to these the mRNA structure. Specifically speaking, the nucleotides that form the mRNA are not they won't just pass through, right? They're too small. I mean, they're too large, right? And their polarity is in several places in conflict with the polarity of the phospholipid bilayer inner nuclear membrane and outer nuclear membrane. So they must pass through these nuclear pores right here. Okay, when mRNA passes through these nuclear pores, then and through whatever region they do, then they finally, what happens to them? They get picked up by a ribosome, right? Either a free ribosome, or these are ribosomes, free ribosome or a bound ribosome. So what's an bound ribosome? An bound ribosome is, as it's named, an bound ribosome is one that's attached to the rough endoplasmic reticulum. So what is the rough endoplasmic reticulum? So the rough endoplasmic reticulum is a continuation of the nucleus. That's how it's typically termed. So it's also a membrane-bound organelle, which has an inner space called lumen, and its outer surface, which is phospholipids, have transmembrane ribosomes. Okay, these transmembrane ribosomes allow, or what do they allow? What is the purpose of a ribosome? Remember, a ribosome facilitates RNA translation. It is one of the fundamental components of life. Okay, you know, a ribosome is probably the most important. I mean, and the DNA is important, obviously. But it's equally important because you couldn't actually build the products from the blueprint DNA without the DNA sending information via mRNA to the ribosomes, and the ribosomes taking in tRNA, mRNA, and synthesizing a protein, right? So basically the mRNA gets read in like a ticker tape type thing, like your old tape machine, and tRNA adds amino acids at the addition site of the ribosome. Now, I'll have to go over all the terminology of the ribosome and the tRNA at some point, and I'll do that just to make sure this is clear. But the endoplasmic reticulum, there are ribosomes embedded into the endoplasmic reticulum. So those are responsible for building proteins, which get transported to the outside of the cell typically. Okay, so remember, the question that came to mind is, why are there free ribosomes and why are there endoplasmic reticulum bound ribosomes? Okay, well, the free ribosomes, your cells need proteins as well, right? So the free ribosomes are typically used, and those are dotted throughout the cytosol, right, of the cell. The free ribosomes, those manufacture proteins which are used within the cell. Those proteins typically don't need complex folding mechanics and other things to be done to them, because those proteins, well, they do in some cases, but the protein products that need the complex folding of the endoplasmic reticulum, and that's what the ER does primarily, the rough ER. Remember, there's two ERs, the rough and the smooth ER. The rough ER is responsible for the folding of proteins and the addition of additional side groups to proteins, right? So the rough ER attaches proteins together as their amino acids. It allows folding of amino acids, right? Of these amino acid polypeptide chains until they get to a part of the rough ER called the transport ER, or transitional ER. So the transitional ER, what does that do? The transitional ER is actually part of the rough endoplasmic reticulum, and that buds out from the rough ER, right? And when that has your packaged protein within it. So because when a protein enters the rough endoplasmic reticulum, it needs a way to actually get out after it's done, right? So once it's finished being synthesized, folded, and modified by the rough ER, then it gets compacted into a vesicle, okay? Vesicle. The vesicle. Now remember, the vesicle, what does the vesicle do? The vesicle is responsible for transmission or a transport of the protein to the Golgi apparatus. Okay, so I've mentioned the Golgi apparatus here. Okay, what is the Golgi apparatus? We have to talk about all of these terms, but the vesicle is another membrane-bound structure that is produced from the endoplasmic reticulum. So you can think of the endoplasmic reticulum, right? You can think of the endoplasmic reticulum as having these parts which are made for excreting proteins from them, right? And again, these form vesicles. These are the vesicles, right? The vesicles are basically globs from the ER, which are formed in order to transport these proteins outside of the ER. Okay, so this is important because the Golgi apparatus, right, is responsible for the Golgi body rather is responsible for the final packaging of the protein. Okay, and what happens in there is that the protein gets modified by several enzymes which are in the Golgi apparatus. And I mean, I don't know right now at this point why that can't all happen in the rough ER, because some additions of monosaccharide units get actually done in the rough ER. I might come back with an answer for that later that obviously everyone has picked up on already, but for now just note that the more significant packaging and modification into final transport vesicles, okay, occur in the Golgi apparatus. And just for shits, let's take a look. Golgi body picture. Okay, so just you want to see what the Golgi body kind of looks like. The Golgi apparatus looks like this, like pancake-looking thing. Okay, that's the traditional explanation for it. It has incoming transport vesicles and outgoing transport vesicles. It also has a cis face and a trans face, which I'm learning more about. Okay, but basically, transport and modification occurs across the stacks of this Golgi apparatus, right? Through each of these membranes, these cisternae, are responsible for modification of the protein, and transport between the cisternae occur through these intermediate transport vesicles, right? And actually, there's a lot of there's multiple theories on how this transport occurs. One is called the stable cisternae theory, and the other is called the unstable cisternae theory. Anyway, not getting too far off track. Finally, what happens is that the vesicle leaves the Golgi body. The final protein vesicle leaves the Golgi body to then leave the cell, because its phospholipid bilayer allows transport out of the cell through various mechanisms, and therefore the cell has excreted the proteins it's meant to excrete. So cells excreting proteins is important because, for example, your liver cells do a lot of this. And all of your cells actually excrete proteins, but different cells have different purposes. So I didn't even get into the smooth ER. Okay, the smooth ER, I'll do that next time. But let's talk about eternal life for a second. Actually, I'm about done with this video. Eternal life. Let's talk about telomeres. Let's see what's up in the news with telomeres. Okay, I want to talk about that. Interacts with to prevent transcription-mediated DNA replication fertilization molecule. See, okay, 17 days ago. How worthy of news do you think this is? Molecules identify that reverse cellular aging processes. Which molecules are those? And what are the specific cellular aging processes Central to a lot of scientific research into aging are tiny caps on the ends of our chromosomes called telomeres. A lot of people know about these tiny caps called telomeres. These protective sequences of DNA grow a little shorter each time a cell divides. By intervening in this process, research hopes to one day regulate the process of aging and the ill health effects it brings. A Harvard team now offering an exciting path forward, discovering a set of small molecules capable of restoring telomere length in mice. Okay, it's capable of restoring telomere length in mice, but under what conditions? Okay, that's very important. Telomeres can often be like the plastic tips on the ends of our shoelaces. Another common example, people always compare telomeres to shoelace tips, preventing the fraying of DNA code of the genome and playing an important part in the healthy aging process. But each time a cell divides, they grow a little shorter. They have to grow a little shorter. And there's a really weird reason for it, but it seems like a mechanical reason that can be circumvented. Anyway, I'll get into telomeres later. This video is already too long. Hope you learned something. See you later.